Hello class. This I believe is where we left off our last lecture. So we're going to keep on going by talking about lipids. We're going to start by talking about their structure. Actually, first by defining them. So a lipid is an organic compound which is a long chain of fatty acids that are insoluble in water. So we learned a little bit about these, but our monomer, or our smallest piece that make up a lipid, is a fatty acid. They are made of hydrophilic polar heads and hydrophobic nonpolar tails. Hydrophilic polar heads and hydrophobic nonpolar tails. So they align very nicely. These tails that are hydrophobic naturally want to be next to each other because there's less water in between them if they're together, while these heads, which are hydrophilic, will be attracted more towards the water. So lipids form these nice bilayers very easily. And so this is our barrier of the cell, with the outside being up here and maybe the inside of the cell being here. And yeah, they're very important for cell membranes. So also an important thing to note about the structure is that membranes can be made more porous or have more holes in them due to our unsaturated fatty acid tails. So a saturated fatty acid tail looks like this kind of molecule. We have a bunch of single bonds between each of our carbons all the way through. Just like this looks very similar to our carbohydrates, but this end is just a little different. And at one side, to make the head, will be a phosphate group. But this is just the tail part, just our tail that is this bent kind of Z shape. Anytime we have an unsaturated fatty acid, we have a double bond. So there can be lots of double bonds or only a few, but they create a kink and change the way the tails look. So we can see saturated lipid tails and unsaturated lipid tails in between here. And as you can see, there's a lot more space in between for molecules or water or ions or any kind of material to go through this membrane versus this one. The function of our lipids, our main function is our barrier. That's what we just learned last time. I should say last unit was the main function of a lipid is the barrier of the cell. So it still acts as a barrier. It helps with cell signaling, and it also helps in energy storage. So because of these long fatty tails, there's a little bit of energy storage. Because we have a few proteins on the outside, there is some cell signaling, but its main job of a lipid is to be a barrier. And I should underline those, so give me 1 second. Awesome, so those are now labeled because we should be writing these down. They're very important. All right, the last macromolecule we have are nucleic acids. So they're made up of nucleotides. It can be either DNA or RNA, and there's 3 major parts. We have a sugar molecule, which can either be our deoxyribose or our ribose, a phosphate group, and then our nucleotide bases. So this deoxyribose or ribose is the part that makes up the D or the R in DNA and RNA. And it's important because it changes if there's an oxygen or not on this little sugar molecule. So this is a sugar molecule. Our deoxyribose, I think, would be missing this sugar right here, and our ribose has it. So I think so. Yep, ribose. So that changes the chemistry quite a bit because oxygen is very electronegative, as we learned about during our water chemistry section. And then our phosphate groups are really important as these nice binders between one molecule and the next. Our nucleotide bases, we have really 5 different ones. For our DNA, we have adenine, guanine, cytosine, and thymine. And a lot of time that's when we see our DNA just in these abbreviated letters of A, G, C, and T. And then for RNA, the only thing that changes is this uracil. So we go from a thymine to a uracil. So GACU. So One way to know if we're looking at a sequence of RNA or DNA is just to see if it has U's or T's, and that can help us. And some more important notes that I want to expose you guys to is that we always write our nucleic acids 5' to 3' when we're writing our letters out. So what I mean by 5' to 3', we even have this nice little direction, but this sugar shows us what I mean. So this carbon molecule is our 5'. We start counting our carbons as 1, 2, 3, 4, and 5. So this carbon molecule is our 5'. This is our 3', meaning this is our third carbon when we start counting, and this is our fifth. So we always write them from 5', which to 3'. Is left to right is 5' to 3'. So our example is here we have our 5' end, so it'd be T, C, A, G. If we were going to write this other one on the right side, we would start on this end and say C, T, G, A. So that's just a good trait or a good thing to get into the habit of doing, because otherwise we'll get confused. Because if we wrote top to bottom, well then what's the top, what's the bottom? It's very easy to notice which carbon is the 5' and 3'. So that's why we do it that way. Also, when DNA is made, it's always made from this 5' end to the 3'. So another reason to write it in that way. Our function, our main function is our information carrying molecule of the cell. It's pretty straightforward. That's what DNA and RNA do. That's why they're there. It's our cookbook of the cell. It tells us how to make things. It tells us what things we're going to need, all of our ingredients, and directions on how to make what we need, whether it's proteins or different organelles. It's incredibly important for that. And then this last slide is just simply a summary of what we went over. So it's really important to follow the whole presentation and take notes as we go, but this is our nice little review summary. We have each monomer, which is what is the smaller unit of our macromolecule. So monosaccharides become carbohydrates, amino acids become proteins, fatty acids become lipids, and nucleotides become nucleic acids, and then their important functions. Here's a visual representation of each one of them for us more visual learners as well, so that we can learn and look at each different way. So I encourage you guys to kind of use this, come back to this as a good review, especially as we're coming up on some more tests. But I hope that all was clear, and if you have any questions, let me know.